model, you can actually improve the density in each of these cycles, and thereby you can obtain better atomic models. So this is an example here of such a density taken from the EMDB model challenge here. This is a peripheral region, so this technique is particularly good if you have resolution variation, which is something that happens a lot in cryo-EM maps. So you see here these areas are largely over-sharpened. By using a model-based sharpening approach, you actually recover very well the actual density, and we noticed that under using such a refinement target, it actually turned out that this conformation was incorrectly placed there simply because it was impossible to see that using such maps. And so this is quite a useful approach that already quite a few people in the community have used, and also given us feedback that this is a very useful tool. So these structures that we determined ourselves, so they are collected in Heidelberg. And this is, we have 2 Titan cryoses by now. You've seen in the video yesterday from Rainer the latest edition, and so altogether we have now 2 Titan cryoses attached with direct electron detectors. Also in the new delivery just 2 months ago was the Thales Arctica, a Polara microscope, and a T12 essentially for screening for preparing samples. So right now this is used by the research groups in the unit, but also we have been running kind of a prototype service platform with the iNext EU-funded grant. So this allows you to actually apply for beam time, and mostly we've been providing high-end data collection. So that means you already have an optimized sample, and this you ship in, and basically people are present, and then we collect for 1 or 2 days as much data as you can. There's also such a thing called enhanced support, and this is something where you can actually have your biochemical preparation and then optimize your sample, but obviously this is much less defined and can take much longer. And then also there's correlative light and electron microscopy that we offer through this platform. And although it's an EU-funded project, it is open to researchers worldwide, and so also you can in principle apply for this. And the good thing about this is actually that it provides accommodation fees and also no measurement time. But it is a peer review process. So in this way, within the last year, essentially, when we started this, we already had approximately up till now, including ongoing projects, 30 projects and 60 days of beam time. And now with the arrival of the new microscope, we will increase this number significantly. So there is another activity that you might be interested in, and you've also heard it in the context of the India cryo-EM course. So we have been organizing a cryo-EM course in Heidelberg. So this is one of really the earliest courses that has been available. So in 1982, this was organized by Jacques Dubochet and colleagues, and then there was a few others that were in Heidelberg, Stephen Fuller and Werner Köpmann, that were also, amongst others, organizing this. And then when John and I were there, we took over this course, and this is also a panel of international people that are actually advising on this, and so this is how we set up the schedule and also planning the actual practical time. And right now we are in the process of planning the next course in September 2018. Yes, so and with that I'll basically have a few more slides on our own research scheme on structure of autophagy assembly. So just to give you a brief idea, so we are working on trying to understand how such small molecules give actually rise to such large independent organelles like the autophagosome. The autophagosome is a double membrane vesicle compartment basically that forms in the process for the intention of removing large cellular structure to the lysosome. And then one particular area that we've been interested in is in trying to understand how cargo is recognized. And this is managed by so-called autophagy receptors that, for example, recognize organelles, also aggregates in other structures. And one of them is depicted here, and it's called P62, and it recognizes an LC3 molecule on the membrane, and then, on the other hand, ubiquitinated cargo that has been marked or labeled for destruction. And interestingly, this P62 molecule is quite important, as it's also involved in many of these protein deposition diseases that can be in the brain, but also in the liver and muscle and other tissue. And for a specific portion of the protein, we resolved a helical structure. So this is now a 3.9 angstrom map of the NBR1 PB1 domain. And I won't really go into the detail, but obviously at this resolution you can build atomic models and you can learn about how these the interfaces are built up. And we tend to also then complement this with biochemical data wherever possible. Now, of course, one of the questions that we received early on is whether this kind of assembly, that it forms such a regular helical structure, was quite a surprising finding, whether this is something that's actually relevant inside of cell, or is it simply an in vitro artifact. And here we turn to correlative light and electron microscopy. So we basically induce the deformation of such so-called P62 bodies, and you can label them, and by this characteristic pattern already you can navigate to the area of interest and then image them. And this is just a regular plastic section EM approach, but already in that you can see, if you now, for example, take tomograms, that a lot of these structures are they are reminiscent of filaments, or they are certainly not like a microtubule, but in this cluster of protein, you can actually identify here certain stretches and also evaluate them, roughly. And this is the width of these kind of structures corresponds very closely to the in vitro assembly. So there's good reason to assume that these filamentous structures that we have solved in vitro are also relevant in vivo. And we believe that such a multi-scale approach is really very beneficial in order to understand cellular processes. And we've done this in another case also for a so-called CVT pathway. This is a particular type of autophagy model pathway, in a way where you can solve atomic resolution structures and do biochemistry and also resolve, put that into context of micrographs from the cell. And ideally what we would like to do in the future is to really zoom on in this area and go further in the molecular characterization, for example, using subtomogram approaches. And here I'll just mention another group leader in the unit who's been pretty successful in doing that. This was Martin Beck, and so I also put his papers in a way on the nuclear pore complex, a bit into the historical context. So during his PhD, he determined an 80 angstrom resolution map from really just a handful of tomograms, and then really a limited number of pores that he could use, 260, and he obtained this relatively crude map. And in his first years of his group leader tenure, he then also managed to push this already just by using a much larger computational efforts to 30 angstrom, and then more recently this resolution was pushed to 21 angstrom. And since there were so many crystal structures that have been solved in the field on the nuclear pore, he could actually propose really an entire pseudo-atomic model for such a vast really molecular complex. Now this is of course also, this wasn't done in situ really, so this was still done with purified pores, or rather nuclear envelopes. And we have now at EMB Heidelberg, a new group leader has just started, Yuya Mahamid. She joined from the Baumeister department, and she is now bringing the technique of focused ion beam milling, cryo-EM to EMBL. And so this is just an outline of the approach. You grow your cells on these grids, and then you apply an electron beam or a gallium beam that actually really removes all this thick material, because the electron beam wouldn't be able to penetrate that. And what you leave it with is a thin lamella, and that is then thick enough to be penetrated by the beam, and then you can get these type of images. And this is a very nice example of her work in the Baumeister department with some ribosomes and microtubules and all these things you can really see in particular detail. And I believe this is certainly something of high interest. There are so many applications for that in the future, and this is really where structural and cellular biology will really merge. Yeah, with this I'm pretty much finished. Yeah.
Thank you, Carsten. If anybody has a very burning question, we can have it now. There's one? Okay, quick question. Yep.
So given that in CLEM you always, or traditionally you always tag a protein with the fluorescent protein, which is most of the cases very highly stable protein, do you have any concern about any potential artifacts?
I guess that's something definitely to worry about. I mean, the moment you attach something, and I think there are now also examples of that attaching a GFP is sometimes required to just localize the protein, but again, it will already affect your ultrastructure there. So that's clearly a concern, yes. Yeah.
So are there other kind of methods coming up, methodologies without using GFP or?
Not that I'm aware of. I mean, I guess there's too little examples of this type of work to really criticize it at that level, because the insight is so new. But I guess what could be done is just to use smaller fluorophores, maybe by chemistry related process with non-natural amino acids, for example, also, where you could circumvent that type of issue. Yeah.
This has obviously been a problem in cell biology for many years, and the general approach is to make sure that you check your localization or whatever results using different tags, or, if possible, an antibody against the native protein with and these kind of checks are always necessary to make sure that the tag isn't affecting things.
Okay, we can continue the discussions during the tea time. Let's once again thank all the speakers for their wonderful talk. We will have a short tea break, and we will. How much, Shelja? No. I think we should. We had such a long session. We will have. Okay, so we will meet at 4:25. Yeah, 15 minutes tea break. Yeah.
to be sustainable training. It's not just one phase or two phase. I think we should really keep in our mind that training means for long-term planning of the training, not just one training session or few training sessions. And of course, for me, the dream is to have a facility, a joint facility in India where we also be equally involved in setting up the cutting-edge infrastructure, carrying out the research, and if we can do that in partnership with the EMBL, that or with the EMBO, that would be really good for all of us.
Thank you, Radha. Natesh comes from ISER Thiruvananthapuram.
Hello. So, I'm Natesh. I'm from Indian Institute of Science Education and Research, Thiruvananthapuram. This is our campus. Just to say that we are in a beautiful campus in midst of forest and mountains. And in the next slide, you will see. Where is the? I think before this, there is one more slide. Oh, amidst this forest, we have this facility as well. We have next slide. We have a 120 kV TEM, and we also have a Bruker rotating anode generator for our in-house X-ray crystallography experiments. Mainly, I am a crystallographer who actually learned cryo-EM on the way in Birkbeck. So, next slide. So, amidst this work, we can also enjoy the beauty. We have several fountains within our campus, at least 4 or 5 streams like this, and we also have a river flowing within our campus, actually. And next slide. So, to basically introduce me, I did my PhD at Indian Institute of Science, Bangalore, and postdoc at University of Bath, UK. And during my postdoc and PhD time, I was a crystallographer, extensively worked on protein crystallography. In University of Bath, I had used extensively BM14 and also P13 beamline in EMBL Hamburg. And during my postdoc, I worked on angiotensin-converting enzyme, one of the important protein in cardiovascular disease. And after my crystallography schedule, one of the key aspects is where we want to train a trainer. One of the best important thing which we should keep in mind is cross-disciplinary fellowships, something which EMBL and DBT can put together and devise something like. For example, I was basically physics background. I learned molecular biology under HFSP fellowship, and then I learned cryo-EM under what is known as Wellcome Trust Advanced Training Fellowship, where I went to Birkbeck College and I learned cryo-EM, and there I did work on single particle cryo-EM with chaperone in non-native protein. In particular, we did a complex with Rubisco, and then we saw the structure with various different classes and averages and states of Rubisco bound to grow EL. And this is the work which we are currently doing at ISET Trivandrum. We mainly focus on transcription regulators, in particular MTB transcription regulators. We are interested in some of the interacting partners of RNA polymerase at the transcription elongation complex, which is basically a big protein complex. Our aim is to do crystallography and cryo-EM. Apart from that, we also work on chaperone non-native protein complexes, which I have worked on ternary complex after coming here to ISET Trivandrum. Binary complex was done in Birkbeck, and this is our team in ISET Trivandrum. Mainly the students who are sitting adjacent to me are PhD students. One girl in violet is another PhD students. Rest and all are the floating population, basically undergraduate students. And one of the important aspects which I want to highlight in this is that undergraduate students are very exciting, and they do lots of projects which are short-term though, but very effectively. In principle, one of the students is in MIT now, who is doing a PhD program. And these basically students contribute a lot to our projects actually in short-term periods actually. And training undergraduates may be one of the aspects which EMBL can take into account. EMBL, for example, we encourage these students to go out for projects every year in the summer for 3 months, and which can be, we can do something where these students can come to EMBL and get trained for 3 months. And these students become very qualitative students for the final year project and PhD later on. And most of our students, unfortunately, they go abroad. We want to retain them, but that's the reality. And this is the type of work which we have been doing. We are working on some of the regulators which interacts with RNA polymerase. This is a protein called GRI which interacts with RNA polymerase, and we have also done a cryo imaging of this RNA polymerase interacting partner called MSMEC6292, and that image was previous image. That image was taken in Manideepa's lab in IIT Delhi, which is actually a very preliminary data. But afterwards, my students, since we did not have in-house facility and I could not go to Delhi again and again, now we have in-house facility, so I have to reactivate them in the last 3 months. It's running in the last 1 month. We are working on that. So next slide. Apart from that, these regulators are also shown to be post-transcriptional regulators of protein quality control. These proteins act like chaperones. So we have characterized some of them through CD and biophysical studies. Next slide. ANS binding studies and next slide, and CD studies, and shown that they are actually chaperones apart from the transcription regulations. And they rescue some of the proteins like alcohol dehydrogenase in the next slide. We see that this protein actually rescues the alcohol dehydrogenase in a dose-dependent manner in protein folding assay.
Naresh?
Next slide.
You can wind up now. Time is up. Yeah.
So we can go. So this is another project which we are working, which is actually a transcription repair coupling factor, which actually expresses in 2 forms actually. One is the hexamer and monomer forms. Next slide. And we have crystallized this protein in the monomer form, and by ab initio method, we have extensively used BM14 and Soleil Proxima 2A, and then Diamond. We did in 2012 the first remote data collection from Diamond for this project. And we have recently solved the structure by selenium derivative. Next slide. So I will not go into the details. This is the structure. Next slide. And we have a ATP complex. This data was collected in-house, and we have the structure with the ATP bound to it, and we see some conformational heterogeneity of this with respect to bound and unbound form. Next slide. And that is the conformational heterogeneity that we are seeing, which is in blue and yellow. Next slide. And so apart from this, the hexamer form, we have actually done a negative stain EM. So when I rotate this hand, you can, next slide. We have the negative stain EM of this hexamer form, and then we actually did a class average. Surprisingly, this is an undergraduate project student who did this project actually, who was a minor from chemistry. And when we did a reconstruction of this, next slide, we surprisingly see a 12-mer rather than hexamer.
I'm sorry, but this is a discussion session. I think we've had 2 talks where the people took the opportunity to make use of the discussion session to say what would be useful in the context of the partnership.
Yes.
It's not meant to be a detailed description of all the science you're doing
Next slide
in the
will
lab.
come with that partnership details.
But really, you've had much more than 5 minutes. You were asked to speak for 5 minutes. We have many speakers who are going to go on. And it's really very rude to continue talking after the chair has asked you to stop.
So I'll stop.
If you want to have something to say with regards to the collaboration.
Next slide has that. Can you go to the next slide? Next slide. Next slide. You can pass on till I say stop. Can you pass on? Pass on to the end. There are some more slides at the end.
You can just say it. I mean, what you...
So those points I have noted down in the slide. And it's basically some of the training program which I wanted to emphasize on, in which we would like to concentrate on getting the people linked together, especially at the PhD levels. If you can link the people together, that it will take as a course of interaction over the long term.
Sure, thank you.
Apart from the workshops.
Thank you, Nitesh.
And there are more points, but I will stop there.
Sure, thank you. So the next panelist is Manideepa.
Hi, I don't have much to add to what some of the other people have already said. Obviously, we're looking for a very fruitful partnership which works for both partners, that is for us as well as for EMBL. And what we are looking forward to is, again, as people have pointed out, access to high-end facilities, which we may not have in India or we do not have in large enough quantities. But for that, also from the Indian side, I think what is very essential is that, well, speaking of cryo-EM, which is what I can talk about, is essentially these high-end facilities are not meant for screening. So what we also need to do from our side is to ship good samples so that good data can be collected at these facilities. But that is essential for us. Also access to, as some other speakers have pointed out, protein expression and purification facilities and drug screening, inhibitor screening facilities. NMR, I think Dr. Neil is here, so he can mention that later. So access to these facilities would be extremely useful for us. And also, as others have pointed out, training programs, because EMBL training programs are very well attended and very nicely organized, as we all know. So it would be advantageous if for our postdoctoral students or even for faculty, it is possible to organize some kind of training program so that we can go and learn new techniques and implement it in our laboratories. As well as I think it would be essential, or it would be very good if later on we can think in terms of setting up joint facilities in India, which we can also contribute to. Another thing in terms of manpower, which I was thinking of, as we have a good population of undergraduate and PhD students, undergraduate students usually have their summers free, so they can go to EMBL, learn something, and then contribute to EMBL specific projects as well, so we can provide some manpower in that respect. So I'm really looking forward to a fruitful collaboration over the years.
Yeah, thank you, Manideepa. Deepak? Just if you have some additional...
One second, let me... Sorry to interfere. The way it was planned, I think there's maybe some typo in the program. The speakers who already spoken, they don't need to add anything more. And the panelists who had not spoken, the ones who speak something, and now we keep the floor open to questions. At least the audience will get some chance to say something, and you are the ones who are going to answer to the audience, whether Indian context or from MBL context.
Also, if you have any other specific point to make, one can take it up. Otherwise, we can open it. Deepak, you have something we can take it to the floor. What about the other speakers?
We are fine.
Is it fine? Yeah. So I had a question for Carsten. You talked about this atomic model-based refinement in the cryo-EM map, which improve, correct?
Yes.
Is it something similar to this histogram matching approach which we crystallographers used to do some time back, or how is it fundamentally different in terms of...
Yeah, I didn't go into the details. So it's
quickly,
not based on histogram matching, it's based on calculating the local amplitude fall off, and that's being used to scale this local part of the density map. And that, because this is a major problem so far, that the amplitudes in cryo-EM are very poorly determined, actually. So the phases are well determined, but the amplitudes are very poor. And that actually gets around a lot of the issues that you have in interpreting what is a noise blob, what is a real density, and so on, yes.
Now the session is open to the floor for any questions. Neil?
Hi. I'm Neil from ICGB New Delhi. I'm basically an NMR spectroscopist, and I thank the speakers and of course Dbt Salja and others for bringing us together here for a very fruitful talks and discussion. What I see is actually from the Stephen's talk that NMR facility is also in EMBL. So is there any plan that NMR facilities like high field spectrometers, which are not there at last, in country we don't have a very high field spectrometers, 900 and above. And specifically, they are the state of the protein, which are the nasty state, the bad ones, the aggregates like Alzheimer's ones, which can be only dealt with the solid state NMR or with the liquid also, because the small one turns into the bad one. So is there any possibility that this NMR facility could also be used by during this partnership program?
Naturally comes into it, but let me ask Stephen.
Yeah, I mentioned NMR in the context of the Grenoble campus there are excellent NMR facilities there, but they're not actually run by EMBL. They're run by the French Institut de Biologie Structurale. They are open as a user facility within the French context, and even at the European level within the Instruct context. How this can be, whether this could be extended to India is up to them in some sense. As I said, we are trying to find mechanisms to open all the facilities on the campus in a kind of coordinated way, but this is quite difficult. And in the particular case of NMR, it's not really under EMBL's control. There are NMR facilities at Heidelberg. Maybe they should be mentioned.
Just another small thing on the other part. Means people have mentioned the training thing. So what I think one way will be like exchange can be also on-site trainings or off-site. But some of the webinars, some of the slides, some of the videos which can be used for, they can be also made accessible maybe on the freely or on the, with the login sites or whatever way, so that the students can, whenever they want, they can look at those webinars or those videos, and they get trained specifically when they are these automated beamlines where they have to put it, all these things. So they have a better experience with that. So those can be also planned. And something also about the softwares which are developed can be also be accessible to in India.
Sure. I think there was some discussion earlier on the bioinformatics platform that they tried to bring in some of that for the bioinformatics operation. But I don't know, do you have any plans for the instrumentation operation as well? Yeah, sure. It allows that possibility, yeah. One can look at, yeah. Particularly when you have long distance partnership, you know, it's not accessible for everybody to be going there, but this can be very useful, yeah.
Yeah, this is an area that's underdeveloped, I would say, from our point of view. We don't have the big training group they have in EBI, but it's certainly an area where we could look at ways where videos of things going on could be put on the web.
You know, particularly when you have a geographically very distant partner, these things can become very useful, actually, yeah. Any other? Okay, Arul? Yeah.
I'm Araki Asami, I'm from I'm coming from ICGP. I'm Araki Asami, and nothing problem with this. My question is, in addition to whatever has been suggested by, including the speakers floor, I was wondering, it's great to access these facilities through like ESR of DBT. So it will be nice to access to EMBL facilities as well. But my question is, can we also it's a suggestion, have a program grants to utilize structural biology tools to come up with translational research, to use the facilities there, and can we come up with program grants that will fund that kind of a research? Say, for example, non-communicable diseases are, we are going to beat all developed nations soon. And like atherosclerosis, cardiovascular diseases, or diabetes, or it could be TB. So can we have specific...
Sure. No, that is something which internally we need to discuss and to see how we evolve such programs so that we can effectively use this partnership.
Exactly.
This was also mentioned earlier, but that's something which we can look at. Yeah, sure.
Thank you.
This is Gitanjali from NIPGR New Delhi. Yesterday's, this morning's, and today's deliberations have been very interesting. I particularly enjoyed your talk, Manideepa, where you were able to describe how the cryo-EM is a small community, and yet together you guys meet up and you're using facilities and providing access to each other. Do you also have, or do you plan to have an online platform where others not from the cryo-EM community could potentially make use of the facilities or the trainings or your expertise for their own work. It is related to what others have been asking, but I'm particularly interested because I'm from the plant background. And I know a lot of friends and colleagues in the agronomy as well as developmental plant biology and plant genomics areas, people who would be more than interested in getting associated with the existing Indian platform, leave alone with notwithstanding what we might or might not come up with EMBL. This is a really good initiative that your community is, and we should probably also have a bioinformatics, structural biology community like this. Communities there, online efforts. So have you initiated any, or do you have any plan to initiate any? That's my question. Thank you.
So that's a really good question. What can we do to engage the broader community in India? So far, as you all saw, the cryo-EM community is very small, although it is growing fast. And the good thing is that we all support each other in whatever small ways we can. To answer your question, whether we're planning to have any resources online. So from the 2016 course that we had for SEM3DIP, all the lectures from all the teachers were put online, and they were also streamed live so that people can actually access those. But I agree that some basic theory and basic workshops are required, because what we do for the SEM3DIP courses, they're mostly geared towards people who already are doing some EM or who are practicing structural biologists. So I do think from your question that should give us food for thought, that we should have, basic courses that we should do online. We should have that information online, which more people should be able to attend. And I think actually the platform for that should be EMSI, the Electron Microscopy Society of India, and the Life Sciences section of that, which we can try to broaden, and the SEM3DIP can be more for more advanced users who can come and learn how to do a reconstruction with their own data set. But your point is very well taken.
Thank you. Thank you, Manideepa.
Should SEM3DIP have a YouTube channel then?
One can get towards that. Right now it's not there.
Okay. So I apologize, I didn't introduce myself in previous questions. My name is Shorojit. I'm from an institute called Bose Institute in Calcutta, and we are running the show for last 100 years, and this is actually the centenary year for the institute. And I'm a Wellcome Trust DBT India Alliance Fellow in that institute. And I asked quite a few questions already, and I have a number of them now, so apologies again. So the first is, we have some high-throughput screening, crystal screening facilities in India, but as far as I am concerned, none of them sends time-dependent images, like at second day, 7 day, those kind of things. If I'm correct, I don't know if I'm correct. But Hoffman Institute in US used to do that. Does EMBL has this kind of service where you would send images of the plates or drops in a time-dependent manner for the crystal screening conditions?
I think Hamburg has such a facility. I think he can. You also have, yeah.
In both Hamburg and in Grenoble.
Okay, thank you.
Yes, but then the question is how one arranges this. Cost.
Okay.
Charge for plates, also internally.
Okay, all right. So the second question is for crystal direct approach. So when you get a sample from outside, do you think that others should have screened the condition already and then send them to you, or what would be the approach?
How this works is that an academic group will have a system where they have a well-defined crystallization condition which permits soaking of ligands. Then they transfer the whole system to Grenoble, and all the work is done in Grenoble, so that the crystals will be grown there, they will be soaked there, they will be then harvested and transferred to the beamline. this is the most efficient way of doing it. So that's how it works. Obviously, this has a limit in capacity. It takes quite a lot of work to do all this, and so it's not open to hundreds of users. Right. And again, there's a peer review system. At the moment, Access is financed by a European grant, which you've heard about, this thing called iNext. And again, we have to find mechanisms whereby this could be extended to Indian usage. And I think in some way that would require some contribution from India, because it's a non-trivial exercise. But I think it's very valuable for those people who are really serious about going into drug design, structure-based drug design.
In fact, another advantage of these crystal direct plates is that you can do in situ data collection. So we have actually done a number of screenings from scratch and then try to find crystals with the X-rays and not with optical light or whatever. Because in the end, the only proof that you have a crystal is a diffracting crystal is that you see diffraction. Right. So that's a route that we really want to go, and then we would simply start from scratch for systems.
Right. And I have another one.
If anybody else has a question, if not, you can have one more. Okay, go ahead.
Okay, so again, it's a sample prep thing. It's for cryo-EM. So if one has to use the high-resolution systems in EMBL, so would you prefer them already having, say, at least not having preferred orientation kind of thing, problem in their sample or in the grid?
Yeah, okay, now. So the way this works, I mean, normally, obviously there are very different levels of when people step into the process. So optimized in that moment means homogeneous monodispersed particle population, right? Thin ice and so on. And whether it's preferred orientation, you may need to figure that out. And obviously that may then require a further round of refinement. I mean, that's how it goes. I think it will be hard to make these kind of very, very specific requirements. I mean, I think we can state as far as we have thin sample, homogeneous, a reasonable number of particles. I think that's what we would normally think of as optimized, yeah.
Right.
Sashi has a question?
No, I don't have a question. In case this session is over, then I want to start.
Please.
So, right now in ESRF, you can submit a cryo-EM proposal in a rolling mode, and what it requires is that you submit good images of frozen particles along with a 2D classification and a 3D model, if that is available, to show that the data is good and to justify that you can collect it on, why would you want to collect it on a high-end machine?
Yeah, so maybe I thought I will ask Stephen to possibly comment on this or suggestions on this particular topic of how, with the earlier model of collaboration that you have had and interacting with a larger community, how you think that you would like to, take this forward in terms of a larger collaboration of multiple facilities open to an even larger community. Do you want to? That will help us in terms of developing whatever platforms that we need to have here in terms of benefiting from this collaboration. See, earlier we have had a successful model for, synchrotron usage. Now we are talking about multiple things starting from imaging to bioinformatics to different platforms in structural biology. And what's your suggestion in terms of, taking this forward?
Well, at different aspects. We talked a lot about training and workshops. That's one thing. But I think collaborative research projects is going to be very important. And there was some model from other associate member states where that's like Argentina, where the associate member state had some funds which allowed postdocs and students to go for a limited period, 3 months or so, to work directly in an EMBL lab. And I think some such mechanism as this is going to be very important to give really close, collaborative projects work. And that's something that has to be thought through. And then that has the advantage, it kickstarts collaborations that may then prolong or develop. It gives the person who comes training and an inside view of how things work in a high-level lab like EMBL. And I really think this is the thing that has to be developed.
Thank you. Aarti? Quickly, yeah. Sure. Use the mic, use the mic.
Since you gave an example of a success story in this case, who initiated that? Was that initiated by PIs?
I can give you a very concrete example because this was, again, in relation to Argentina, another of our associate member states, where the Argentinian government provided funds for this kind of insertion at EMBL. And at Grenoble, we had a person who wanted to solve the structure of a particular crystal system. It wasn't a subject area where we were scientifically involved, but technically we could help him. And he came over, got involved with all the crystallization, structure determination, with the help of our local experts, and solved his structure, went away happy and with a lot of more expertise and with the connection to EMBL. So it doesn't take a lot, but it has to be funded in some way.
Thank you. So if there are no more questions, we will thank all the speakers as well as the panelists for a wonderful session.
So thank you very much to all of you. What we'll do now is we'll sort of have a summary from all the 3 sessions. Now we'll count in seconds, not minutes. So we'll start backwards. So structural biology, 180 seconds, and then we'll have bioinformatics by Partho in 180 seconds, and imaging I will do in 120 seconds. And then myself and Ian will say a few words about the summary of the entire discussion and the roadmap. And then Shailaja will...
Sashi, I will save you more time. I will take just 5 seconds. Whatever the discussions that we have had, I have actually made a summary out here. I think this gives you some kind of a roadmap for what we possibly want to do.
Just today?
Exactly, yeah.
Wow.
Yep.
So you had a premonition?
Kind of, yeah. Because we already discussed with all of them through email as to what they are looking forward to. There are a couple of things that came during the discussion at this point. See, one was with respect to the videos of these technologies, particularly when you have geographical distance of partners like this, that can be a very useful addition, in my opinion. I don't remember any other point that came in addition to this. As Shashi said, there was a premonition, but if I have missed something, one of you can point out. Deepak? Deepak is just, yeah, is there anything that we discussed now that we have missed from this? Okay, as far as I remember, that was an additional point. Sure. Thank you.
2, 3 themes that sort of emerged during the bioinformatics discussion. One's, of course, training, and the EBI is very geared up to training, and so it'll be quite easy to implement that, I think, depending on our ability to get on as or engage with them as an associate member. Having said this, there is also, it's also in the area of bioinformatics, it's also easy to scale up, and scaling up is absolutely essential because every biological science is becoming very quantitative and very data rich. So there will be more and more demand for these kinds of people, and that's not too difficult to scale up. I mean, bioinformatics is an area that's not too difficult to scale up. The second is access to technologies, and we have heard about the cloud system that's already accessible for us, for many people, And secondly, many of the software packages of the EBI are, of course, in daily use by some of us. So that's already accessible in some ways to us. And like one of the questions that was asked in the morning, could we submit our data and run the software packages? That's not going to be easy as of now. That's what we have heard because of security issues. But many of these packages are downloadable, and if we are able to access the embassy cloud, then that will be, we are already able to access. So in terms of access to technologies, bioinformatics, EBI already has made some facilities available to the community. In terms of scaling up of the training programs, that's easy and doable. So it's all, I think what we need to do in India is to sort of, some people should take the initiative and engage with EMBL, EBI in order to facilitate bioinformatics. And that's probably easier than the other two areas, but that's my sense of it. So these are the other kinds of things like, engaging in PhD programs, having visiting scholars, visiting professors come to India and spend some time. All of those will take a much longer time to negotiate and bring to the table. But I'm hopeful that, once we make a beginning, then these things will follow. Thank you.
Thanks. So, as far as the imaging is concerned. So, as far as the imaging is concerned, there's a sort of the discussion sort of led to more on the training component, is more than the use of facilities, the way the structural biologists use facilities in Europe, whether EMBL, ESRF, Hamburg facilities. The reason is there is reasonably good imaging platforms in India. I think it's more about the training to use those facilities. We need 3 different levels of training. One is large-scale training, maybe on image analysis, but a high expertise group, and who can actually do image analysis not only for Indian researchers, also for anybody in the world, more like an outsourcing method. And also the image analysis related to diagnostics can be developed with the help of this kind of training. The second component is the large-scale facility management, both India and as well anywhere in the world. That also includes technology development. So some kind of workshops we organize regularly in India on technology development, and some of the trainees of these technology development workshops can be potential employees for the companies, microscope companies. Particularly in India, large number of microscope companies are facing problems. Large number, there are only 4 major ones. If you take the Leica, Zeiss, Olympus, and Nikon, facing severe crunch of specialists. They keep looking for technical people. I think it's also academic community's responsibility to provide manpower for them, human resources. And also we need to create some kind of opportunity for interaction between the in charge of the facilities in India and in EMBL through some kind of an internship program. Particularly the mid-career level ones, not the entry ones, who have already have the large experience of running facilities, and also go to EMBL, for example, and then be part of their facilities for some time, and then come back, and this also establishes international linkages. So these are the few salient points of the discussions of yesterday's session on imaging. Okay, so again, to take forward what Vijay and Ian were discussing here yesterday, what we are planning to do with EMBL is a partnership, is to first bring the community together through one more channel. We have a associate membership of EMBO, and of course, individually scientists are collaborating, interactions, maybe attending the conferences together and meet. And of course, structural biologists have been using some of the facilities of EMBL in different places. But this is to further intensify those interactions, number one. Number two, create some kind of capabilities in India to further high-end research in the area of bioinformatics, image analysis, bioimaging, and structural biology. And the third is to generate human resources of very high expertise, human resources for the world, not just for Indian community, and also for the rest of the world, taking the demographic dividend of India. And that both for industry and for academia. It's not just for academia, it's also for industry. And for this to happen, we have to work together, and the community has to own this on both sides. And we are very happy, heartening to know the best of the EMBL scientists, best of the core facility managers are here. And that shows the seriousness in which EMBL has taken this, and we would reciprocate the same way. And we also have the best of the young scientists of Indian community are here, and that also is very nice. Which one?
Because the proceedings are being webcast
Oh,
live,
yes. Oh,
we
fantastic.
have 150 concurrent viewers at a given time.
So this whole proceedings is being webcast, and what Shailaja is saying that at any given time about 150 people are watching this. That also shows the seriousness. Thank you very much for all the participants on web. So so what we need to do is, of course, based on all the inputs, some kind of partnership agreement will be drafted, and definitely there will be workshops, training, interactions, use of facilities will be there. In addition, there will be a hopefully partnership program in which there would be more like yesterday's discussion on a particular theme. An interinstitutional partnership will be developed, interinstitutional within India and also with EMBL. And that theme will then sort of cover all the three areas: imaging, structural biology, and bioinformatics. Bioinformatics and image analysis and structural biology and bioinformatics, you cannot separate them. So in a way, all three are integrated, right? And with cryo-EM, your structural biology and imaging are also integrated. So I guess these three components of partnership is fantastic. And to add to India's strengths, in addition to emerging scientific community, large number of young people, the education in interdisciplinarity in terms of their computational skills and the physics and mathematics and chemistry they bring in on the platform. There's one more large investment is being made in India, which Alok mentioned. I thought I want to reiterate that. A couple of €100 million are being spent to increase the supercomputing capabilities in the country. Large number of supercomputers are being established, somewhere between 500 teraflops to close to about 1.5 petaflops. So in about 5 or 6, that kind of supercomputers are being set up across the country, one in Pune in our institute, one in Bangalore in the Institute of Science, then one in IIT Delhi and a few other places. And all these will be available for biologists to process their data. So when you want, for example, the routine image analysis, Rainer mentioned that a very good STED microscope or something will generate close to about 1 terabyte of data for just one image, one sample. So that kind of image analysis, if you have to do, you really need supercomputing facility. Similarly, these days, all the metagenomic analysis, you need very high computing facility. And structural biologists, of course, do need high computing facilities. And with our investment in supercomputers, we are almost ready to take off. So with this, I think I'll call Ian to say a few words.
Okay, thank you very much, Hashi. And this is, one of the interesting things about being the last speaker is that you go from being a pioneer to being a plagiarist very quickly and very easily. So I am not going to say anything new. I will speak to some of the issues that were raised today, some of which were raised also yesterday, and try to give some details. But what I'd like to set out First, and this is repetition partly of what Shashi has just said, is that the goals of the associate membership, the goals of the association, the partnership between EMBL and India, and we use partnership in a particular way, so I will use the word membership because it means for us something different, is to advance our collaborations, and we've chosen 3 areas. The 3 areas which we've talked about over the last couple of days: bioinformatics, imaging, and structural biology, as areas where both sides are strong. These are all 3 areas that depend a lot on continuing technical advances, and that they are also areas where technical advances are happening very rapidly. We've seen over the last couple of days that there are already strong interactions. That is deliberate. The idea to have India as an associate member is not a completely new one. We've been discussing with DBT for multiple years. And so to engage with the community and to see how well do things work, what works, what doesn't work, has been very useful. The example of BM14 actually predates this, because this was an example which came about because of an opportunity which India, ESRF and EMBL were able to use, but it also demonstrated very forcefully that it was easy to work with the organized Indian community in the structural biology area, and it's not the only area in which you have some organization. So I have to also remember to say that we do want to work together with the top scientists, with the best scientists, but I think it's also important to remind everyone and to remind us that the training activities in particular should go beyond this. We should be disseminating the expertise, we should be disseminating the knowledge beyond the top institutes. The top institutes need access to infrastructure. They are the people who are ready to collaborate. They're not necessarily the people who are most in need of the training, because there are many other institutes who don't have access to many of the things that the top institutes do. That doesn't mean we would exclude anyone from the training. All I'm saying is it should be a broader activity. And of course, we've said it over and over again, the idea is to do things together that are of benefit to both sides. So, interdisciplinary collaborative research projects are obviously the secret to building long-term relationships and building long-term research relationships. Access to the facilities, there will be access to all of the EMBL facilities. There is, in Heidelberg, we have NMR up to 800 what would you call it, megahertz? I can't even, megahertz. Nanohertz, it doesn't sound like very much. Megahertz. And these machines are available to external users. There are people who run them. NMR is not an area, I must say this was a decision we took strategically about 12 years ago, that we would prefer to invest more in cryo-EM and in all of the various X-ray techniques than in NMR, but we still have an 800 machine is still a good machine, and people do NMR every day at EMBL, so this is accessible. In relation to training, bioinformatics, Kath spoke about this, we've spoken about it over and over again. There the model is quite mature. There is quite a lot of e-learning material, and there is a very definite goal to work on training the trainers. There is, within EMBL, an initiative to spread e-learning to the other areas. So this was another of the points that was raised today. That is differentially developed, but you can be assured that we are working on making what will be useful available to the community by internet and by e-learning methods. I repeat the joke I said yesterday, that there are things that you don't learn. And I think, the joke was, it's like learning to play soccer by watching YouTube. I actually, by chance, saw about 50 minutes of the game of India against Ghana last night, and I think this is probably how the Indian team was actually trained. Okay. So the timelines. The next step is that we should sign the membership agreement. This is very close, I think, to being ready. We discussed this yesterday in some detail, yesterday in the morning. As soon as this is signed, then all of the things that I've talked about are ready to go. In addition, and Shashi has mentioned this, there is an intention within DBT that we should also move towards setting up a partner lab, ultimately a network of partner labs, and this will require internal discussion to decide what will be the topic, what's the best area for you to invest in, which is the area that will give you the best return for this. Visitors to EMBL are always welcome. We have a limited number of fellowships for people to visit the core facilities and for people to come to workshops and conferences. Some of those are funded by industry, others are funded by our alumni association, but these are limited numbers. So I hear what you're saying. I think this is a message which perhaps the DBT can think about. How can a program of visitor-funded visits be made possible? But everyone from PhD student to faculty is very welcome. I heard that undergraduates are doing also research here. I think that could conceivably be possible. We have very, very few undergraduates in EMBL, so the number would have to be very limited, but nothing is basically impossible. So that's it. That's all I wanted to say, except to thank you all very much for being here. Thank you all very much for engaging. Thank you of the people who couldn't be here, but who have been watching this by the video link. I hope that you were able to hear and to understand the talks and the questions. I think the audiovisual people have done an extremely good job in keeping things going. But most of all, I would just like to thank everybody who was here, including the ones who are not here anymore, for making the time to come here, talking with us, and getting to know us a bit better, and making new connections, which we hope will form the basis of our working together in the future. Thank you all.
You can't go away without listening to DBT. I'm sorry. But thank you, Ian, for being here, and the EMBL team for coming all this way and spending 2 days with us. This is a culmination of discussions with Professor Vijayaraghavan and Ian for over a period of 2 years. And the 3 themes have been a brainchild of Dr. Vijayaraghavan, and he thought that would really be bring a lot of value to Indian science. And becoming associate members of EMBL will allow almost immediately access to every facility and every program of the EMBL, which every Indian scientist can apply to. Of course, they will have an access only on a competitive basis, but you can apply to all the facilities and the programs of EMBL. The second rung of what we are discussing with EMBL is setting up of partnership labs. And for that, and from what I hear from Indian scientists, is that we are investing a lot on infrastructure. The government, various government agencies are investing a lot in ICERs, in IITs, in various institutions. But I do not know whether we are optimally utilizing the facilities which we are putting up. 100% usage of facilities is one thing. Outcomes of those facilities and the usage of time is another thing. So are we setting the right research questions is one thing. And the other thing which the thoughts which came, are we designing our experiments right? And not all of you. I'm wondering whether it is possible. Is the Indian scientists, do they need senior mentorship there? And can EMBL act as a mentorship program when we have an EMBL partner? Can we then be partners in looking at the larger science questions and solving them together? And this is what we are looking for from EMBL. With the trainings and everything else can be taken care of, but it is the leadership in science that we are looking from our DBT. We are more ambitious, and we are looking for more ambition from our Indian community. Thank you.
Of course, the big thanks to all the staff of DBT who helped, particularly Vaishali, who sent emails to all of you and been here taking care of everything, and all other staff who I don't know the name, maybe Shailaja know the names. But
I really would like to thank Vaishali. It's not just about organizing the whole thing that she does because she has no other option, but because of her involvement in the program, she is completely enthusiastic about the program, and she understands the scope of the program, and she really is going to be the future who will lead the program because she understands the program so well. And for that, Vaishali, thank you.
We can have some tea on the way. So there'll be more